Okay, thank you very much. It's really an honor to be here. I want to thank Dr. Avedon for the invitation and to follow these two illustrious speakers. I'll do my best. I'm going to be talking a lot about mice, which will be a little bit of a change in topic, but I think the topics that we'll cover have clear relevance to what both of the previous speakers talked about. I don't really have any relevant financial disclosures. So, in modern life, our circadian system, which maintains the 24-hour rhythms in our body, is under constant siege. It's basically there's a battle between the way we live and the way our body wants us to live. And mainly that revolves around the exposure to light at the wrong time of day. So this map here shows light at night throughout the United States and Canada. And it kind of illustrates that we are constantly exposed to light when we are not supposed to be. And our circadian clock is entrained by light, and light exposure at the wrong time of day can throw off the circadian clock. Some of the worst culprits actually are these new type of lights illustrated here, these LED lights that are now everywhere. They have a lot of blue light component to them, which is the wavelength of light that resets the circadian clock. We also have our screens. We're all watching TV, watching our phones, watching our computers late at night, and other light exposure. And finally, aging. We're all living longer. The circadian system declines as we age, so there are a lot of people with compromised circadian systems to begin with, and that probably interacts with some of these other issues. So there's an epidemic of circadian dysfunction in our world and in our hospitals. So I think that ICU exposure, this is a particularly bad case it looks like, but there's obviously plenty of literature implicating stays in the ICU and in hospitals in general in poor circadian hygiene. So there are a lot of issues, and I'm not going to kind of belabor these points, but there are a lot of issues in anesthesiology and critical care medicine that relate to circadian biology. Some of them have been touched on, but there's this issue whether or not anesthesia or operations, surgical interventions themselves can disrupt the clock. There's the issues of people coming into the operating rooms for elective procedures or being admitted to our hospitals may have premorbid factors that compromise their circadian function, age being one of them, neurodegenerative diseases being another. And then there's the issue that was just discussed about long-term cognitive impairment after hospitalization and how does circadian dysfunction kind of play into that. I don't have an answer to any of these, but I think some of the mechanisms that we'll talk about may have relevance for future research in all of these areas. So just to review the circadian system quickly, the master clock of the body is in the suprachiasmatic nucleus in the hypothalamus. It's directly connected to the retina, and so it gets light input, and it is the portion of the clock that's directly entrained to the external environment, and particularly the external light cycle. The SCN kind of functions like the conductor of an orchestra. The individual members of the orchestra are cellular clocks in the periphery, in both different parts of the brain as well in different organs. Almost every cell in our body has its own circadian clock system, and all these countless cellular clocks are entrained and synchronized by the function of the SCN. The SCN also directly controls the timing of sleep, and it also controls the timing of certain endocrine functions. So it's a very important nucleus in the brain. Now, there's plenty of evidence suggesting that aging, nighttime light exposure, neurodegenerative pathology, and as I just mentioned, ICU exposure or hospitalization can affect the circadian clock and sleep-wake cycle in many different ways. So the way that we're going to focus on today is kind of at the level of the cellular circadian clock. And so what is this cellular circadian clock? So within each cell there's this conserved machinery that I'll just refer to as the core circadian clock, which is really a transcriptional translational feedback loop that is timed to a 24-hour period. So the linchpin of this is this protein called BMAL1. So that's one that you need to keep in mind for the rest of the talk. BMAL1 is the one gene in the mouse genome that you can knock out which disrupts the circadian clock entirely. So a mouse lacking BMAL1 becomes totally arrhythmic. BMAL1 is a transcription factor that heterodimerizes with either clock or NPAS2 to form a complex that then binds to DNA, these E-box motifs throughout the genome, and drives transcription of genes. Among its targets are the negative limb of the clock, which are reverb genes, period genes, and cryptochrome genes, which then feedback and inhibit the function of BMAL and clock. And so this whole loop is timed to a 24-hour period by a bunch of other factors that I'm not going to discuss. So if you take some cells out of a body and put them in culture and you synchronize them by changing the media or stimulating them, you'll see these very nice oscillations in BMAL. You can see it goes up and back down for the 24-hour period. Even in the absence of any light exposure, this clock runs on its own in cells without any external stimulus. The negative limb genes, such as reverb and period, would oscillate out of phase with BMAL. But it's not just the circadian clock genes that oscillate in cells. It's actually between 10 and 30% of all of transcriptome shows circadian oscillation. And it's highly cell type specific and highly tissue specific. So there are genes involved in metabolism, oxidative stress, inflammation, mitochondrial function, a number of processes that are really important in neurodegeneration that are controlled by the clock. So my lab's really been interested in what does this circadian clock, core clock, do in the brain, in different cell types in the brain, and how might disruption of this clock influence neurodegeneration and neuroinflammation in models of neurodegenerative disease. So just in trying to tie this back into surgery and post-surgical ICU, there was this study that came out in the Lancet that I think is sort of interesting and relevant that showed that people undergoing aortic stenosis surgery, many of you are probably familiar with this study, seemed to do better when they had their surgeries in the morning. So the people that had their surgeries in the morning had a lower risk of adverse events than people that had their surgeries in the afternoon. The authors of this study then looked at gene expression changes between the morning and the afternoon, and the number one gene they come up with that was higher in the morning group was BMAL1. This is the high group, they do better. Another gene that I'm going to talk about a lot today is reverb alpha, which is another clock gene which is driven by BMAL1. So this is reminiscent, this study, of many studies in mice showing very similar phenomena, that the time of day at which certain insults happen has a huge impact on the outcome. So for instance, in mice, if you inject a mouse with an inflammagen, lipopolysaccharide, and cause a massive inflammatory response in the mouse, if you inject the mice at 6 o'clock in the morning, ZT0, when they have high BMAL levels, about half of the mice survive. But if you inject the mice at 6 p.m., all of the mice die. And it's the same dose of LPS. If you do the same thing in mice lacking BMAL, they all kind of look like the 6 p.m. group and they all die, which suggests that the phase of BMAL or other components of the clock has a huge impact in how this inflammatory response plays out in these mice. And this has been shown in many other models. It's the case in a variety of models, from stroke models to MS to a number of things. But the mechanisms underlying this are not really well understood. So what I'm going to talk about today is sort of trying to lay out some potential mechanisms by which the clock can control, in particular, neuroinflammation, but also neurodegeneration. I'm going to talk a little bit about the control of microglia and astrocytes, which are glial cells in the brain that are really critical in almost every neurodegenerative disease. I'm going to talk a bit about reactive oxygen species and oxidative stress, and then at the very end talk a little bit about the aggregation of amyloid beta and tau, which are two proteins critical in Alzheimer's disease that Dr. Black introduced us to earlier. So the model that we use is a BMAL knockout mouse. So BMAL, as I mentioned, is the one gene you can knock out in a mouse that's not redundant, that will disrupt the clock. And so normally, if this is wheel running data, a mouse runs and then it sleeps when the lights are on, then it runs and then it sleeps day after day. If you turn off the lights, it will shift a little bit, but it continues to have a circadian rhythm. If you knock out the BMAL gene, the mouse is completely arrhythmic. It just runs and sleeps with no particular rhyme or reason. If you look at these mice sleep, normally mice sleep a lot when the lights are on, and they sleep then they wake up when the lights turn off and run around. But BMAL knockout mice basically sleep the same amount every hour. There's no night or day to them. They just sleep, wake all the time with no pattern. They actually sleep slightly more than regular mice, which is an important point. So this is not a sleep deprivation model, but it is a sleep fragmentation model because they sleep in these short bouts. Now, those of you that study mice may know mice don't actually sleep the way we do. They already have very fragmented sleep. but in this case, they're even more fragmented. And you can see the same phenomenon in cells. So this is astrocytes that we culture out of mouse brain in a plate that have a reporter that allows us to look at the period 2 gene. And you can see just we give a serum shock here to synchronize the cells, and then the cells show this very nice circadian oscillation. It kind of peters out after a few days, but if you give them more serum, they'll start oscillating again. But if you knock out BMAL, they lose this, and they become arrhythmic even in the plate. So disrupting BMAL gets rid of not only behavioral rhythms, but also transcriptional rhythms in cells. So what we found was mice that don't have BMAL, that have this disrupted circadian system, don't do very well. Their brains don't look very good. So one thing is they have a lot of oxidative damage. So this is a measure of lipid peroxidation called F4 neuroprostanes. It's very high in the brains of these mice. And we think it's because many genes in the genome that are involved in redox homeostasis and redox defense show a nice circadian oscillation. So this is one called NQ01. It's part of the NRF2 pathway. In normal mice, it oscillates. It goes up when they start to become active. In BMAL knockout mice, it's just flat. And so at this time of day, there's inefficient detoxification of radicals that leads to oxidative stress. These mice also have spontaneous synaptic degeneration. So this is synaptophysin positive terminals in CA3 portion of the hippocampus that are lost in BMAL knockout mice. You can see in the electron microscopy, BMAL knockout mice have a lot of these oversized presynaptic terminals that are devoid of any normal vesicles, which are a sign of degeneration. Loss of this circadian gene also makes neurons more susceptible to death. So when we knock down BMAL using a virus, which lowers BMAL levels, the neurons begin to die. You can see these are the BMAL deficient neurons. If we treat them with an oxidative stressor, hydrogen peroxide, it basically kills all the neurons. So they're very sensitive to oxidative stress. And we see the same thing in mice. So we treat mice with a mitochondrial toxin called 3-nitropropionic acid that causes striatal degeneration. If mice have only one copy of BMAL, so they have lower levels of BMAL, they actually have a larger lesion size than wild-type mice. So it suggests that you need a certain amount of BMAL to have defense against oxidative stress in the brain. Oxidative stress is kind of a ubiquitous player in a variety of neurodegenerative diseases. So switching gears again, we also noticed that BMAL deletion causes very profound astrocyte activation. So astrocytes are glial cells that are important for the support of neurons, and they play a lot of roles in the brain. And when they become activated, they express this protein called GFAP, glial fibrillary acidic protein. And in general, that's a sign of unhappy brain. You hit a mouse on the head with a rock, or you cause a stroke, or you give it Alzheimer's, it will get gliosis. These mice get spontaneous gliosis by about 2 months of age, and it goes up from there. So you can see this is a huge amount of astrogliosis in the cortex. We see this in conditional BMAL knockout mice as well, in which the whole brain is basically filled with activated astrocytes. We see less of it when we knock out BMAL only in neurons, suggesting that it may be a cell autonomous effect. We've looked into that a little bit more using some viral models where we can inject a virus into a mouse that has a floxed BMAL allele and knock out BMAL only in astrocytes. So this is a GFAP promoter, so it's an astrocyte-specific promoter driving the Cre virus, or the Cre protein that deletes BMAL. This is the control virus here. You can see we light up nice-looking astrocytes that turn green. They don't look activated, they don't express GFAP, and they still have BMAL. However, in the ones that are given the virus that knocks out BMAL, they almost all become activated, express GFAP, and BMAL is gone. We showed the same thing in an astrocyte-specific BMAL knockout mouse using an astrocyte-specific Cre line, the ALDH101 line, that within a few months of us knocking out BMAL, that these mice develop astrogliosis. We can also see the same phenomenon without using genetic intervention. So if we take mice and put them on an abnormal light-dark cycle, so you basically put them on a 20-hour day. They have 10 hours of light, 10 hours of dark. After several weeks, their circadian rhythms become disrupted, as you can see with these activity plots. And the clock genes, instead of showing this nice oscillation in the brain, this is BMAL, this is Rev-erb alpha, the red lines show they just flatten out, they become arrhythmic. And the mice actually get higher levels of GFAP, suggesting that they're developing some kind of astrogliosis. So functionally, these astrocytes that lack BMAL seem to be dysfunctional. They don't seem to support neuronal health as well. In this experiment, we grew regular wild-type neurons on top of a bed of astrocytes, and they were either normal astrocytes or BMAL1 deficient astrocytes. So the green are the neurons. You can see the neurons grow really well on the normal astrocytes. If we treat them with hydrogen peroxide, we kill about half of them, as shown here in the gray bars. If we grow the regular neurons on the BMAL1 deficient astrocytes, they don't survive as well to begin with, and when we give them hydrogen peroxide, it kills almost all of them. So it suggests that these circadian deficient astrocytes have some sort of deficit in trophic support. So this is an ongoing area of research in our lab to understand astrogliosis is important in traumatic brain injury, stroke, a lot of other areas to see how this impacts kind of disease outcome. We're also interested in another clock gene called Rev-erb alpha that I mentioned that was shown in that aortic stenosis study. Rev-erb alpha is actually downstream of BMAL. It's one of the most circadian transcripts that we see in almost every tissue in the body. This is data from the brain showing that it oscillates really beautifully in the brain. In the cerebral cortex, it also shows this really nice oscillation. And if you knock out BMAL, that's gone, and the levels actually drop about 80% if you knock out BMAL. So you really need BMAL to drive Rev-erb alpha. Rev-erb alpha is actually part of the circadian clock, and it feeds back and inhibits BMAL. So, but besides its function, the circadian clock, Rev-erb alpha is also a transcriptional repressor and a nuclear hormone receptor. And it's been implicated in the regulation of metabolism, inflammation, a lot of other processes independent of its circadian function. So we were wondering if perhaps this is a way that the circadian clock controls inflammation by controlling Rev-erb. So in a paper we published recently, we looked at Rev-erb knockout mice, and we found that, a little bit to our surprise, that they seem to have a really strong effect on microglia. So microglia are the macrophages of the brain. They're involved, they've been strongly implicated in Alzheimer's disease recently by some GWAS studies and some other genetic studies. But they're very important in neurodegenerative diseases. One thing we noticed was that just in regular wild-type mice, that if you look at different times of day, there's actually a diurnal rhythm in the activation state of microglia that just happens normally. And then if you knock out Rev-erb, actually you can see that here by the volume of IBA1, which is a marker for astrocyte, or I'm sorry, microglia, that between AM and PM that there's a difference. And if you knock out Rev-erb alpha, you basically lock the microglia in this kind of activated state all the time. You can see these are some high-resolution, three-dimensional renderings of microglia from BMAL knockout mice. You can see they're thicker, they have shorter processes, and these little yellow spots are they're CD68, which marks lysosomes within microglia. So these microglia are more activated and they're more phagocytic, which is quantified here. They also express a bunch of cytokines like IL-1 beta, CCL2, TREM2, generally have a pro-inflammatory state just by knocking out this clock gene. This is some unpublished data, but it looks as though one of the recent ideas in neurodegeneration is that microglia kind of go rogue and start to eat synapses. Beth Stevens at Harvard has kind of pushed this idea forward. It's been shown in multiple models, including Alzheimer's models, that microglia can kind of get out of control and start to eat synapses. And we see some of this when we knock out Rev-erb alpha. So what these renderings show is a microglia, and these little red spots are synaptophysin, which is a synaptic marker. So these microglia actually have synaptophysin inside of their CD68 positive lysosomes in these three-dimensional renderings. And so this is work in progress, but we don't know if this is because these microglia are just cleaning up damaged neurons or if they're actually causing the problem, but we're looking into that. So why we're excited about Rev-erb alpha is because unlike all the other parts of the clock, it's a nuclear receptor, and there are actually drugs in development to target this receptor. And in fact, one of our colleagues at WashU, Tom Burris, made one of these drugs, SR9009, which is a brain-permeant agonist for Rev-erb alpha. And what we found just in some preliminary experiments is that when we take primary microglia and stimulate them with LPS to cause a large inflammatory response, this is looking at interleukin-6, that by pre-treating with this Rev-erb alpha agonist, we can suppress that LPS-induced inflammation. However, when we try to do the same thing in reverb alpha knockout cells, we actually don't see much of an effect. So it suggests that the drug is, at least in this context, somewhat dependent on reverb alpha to suppress this inflammatory response. We see the same phenomenon in vivo. So in mice, when we treat mice with IP-LPS to induce inflammation throughout their whole body, but they also get inflammation in their brain, that if we treat them also with SR9009, that we can suppress some of that inflammation, looking at IL-6, CCL2, a whole bunch of other cytokines. So we're excited about this. There are both reverb agonists and antagonists, and we're testing those in some of our neurodegenerative models to look at neuroinflammation in the context of neurodegeneration. So we hope that this is a way that we can kind of target the circadian clock pharmacologically to get a handle on neuroinflammation. Ooh, too many. So in the last few minutes, I want to talk about some of the proteins that Dr. Black was talking about and some models of Alzheimer's disease. This is a lot of work that gets boiled down into a couple slides, but we have developed this mouse a new B-mel knockout mouse in which we can give the mouse tamoxifen and cause it to become arrhythmic whenever we want to. So it's a postnatal B-mel deletion. These mice, just like the ones I showed earlier, become totally arrhythmic after you give them tamoxifen. And we've bred this onto either an APP-PS1 model. So this is a model that gets amyloid plaques. So the amyloid precursor protein and presenilin-1 proteins, these are mutant human proteins that cause disease in humans that we put into mice, and they get amyloid plaques by about 4 months of age in this particular form. In this model, actually, Dave Holtzman, my mentor, showed that there's this very nice circadian rhythm in amyloid beta in the brain. And so we're interested, and we can measure this by microdialysis for A-beta, so we're interested in if disrupting the clock will change this rhythm of A-beta and will change the amount of amyloid plaque that accumulates in the brain of these mice. At the same time, we also are interested in tau. So tau is the primary constituent of the neurofibrillary tangles, and we think it's the protein that's really more closely tied to neurodegeneration in Alzheimer's disease. We have mice in which we've bred onto this B-mel knockout background that express a human tau protein that's linked to human disease that causes neurodegeneration. So these mice develop tau aggregation, tau phosphorylation about 9 months of age. About 8 months of age is when we took them down, and they develop pretty severe neuroinflammation at that time. So using these two models, we can look at the effect of disrupting the clock kind of in the two different contexts of Alzheimer's disease relevant neurodegenerative proteins. So long story short, what we found is by disrupting B-mel, we disrupt this normal daily oscillation in amyloid beta in the brain, and we accelerate plaques. So the B-mel knockout mice get more amyloid plaques in their brain at 4 months of age. And we think that this is due to sleep fragmentation. And this is because in this same study, we made B-mel mice that had normal sleep and normal full daily rhythms of behavioral rhythms, and those mice did not get increases in plaques. So it was really only the mice that had disrupted fragmented rhythms that developed more plaques. And Dave Holtzman's group has shown pretty conclusively that disrupting sleep can increase amyloid plaques. So we think that by fragmenting the sleep of these mice, we're accelerating their plaque formation. So it seems straightforward so far, but it gets more complicated in, oh, sorry, I wanted to put in a plug. So some work from Jacob Basak, who's an anesthesiologist in my lab, who's here today. He's actually giving a talk tomorrow morning about how experimental sepsis in the CLP model, the colonic ligation and perforation model, can also accelerate amyloid plaque formation in mice. And I think this is relevant because you could have a situation in which sepsis itself is driving amyloid pathology and circadian rhythm dysfunction is also contributing to that. So that's a hypothesis that we're still looking into in the lab. So if you have a chance, go see. I think Jacob already gave one talk at this meeting, but he's giving a talk, I think it's 7 a.m. tomorrow. So if you're so motivated, I'll see you there. But things get more confusing when we got to the tau mice. So this is unpublished data that we're still trying to make heads or tails of, but it turns out that when we look at tau aggregation in these mice, the B-mel knockout mice actually have less. They have less in intracellular tau aggregation, and particularly the highly aggregated forms of tau. They have the same amount of tau, they have the same amount of phosphorylated tau, but these highly aggregated forms that are positive for thioflavin S, the beta-pleated sheet forms, are decreased substantially in the P3ONS model. Similar, we see a decrease in neuroinflammation in these mice. So as you'll see here, the knockout mice without tau actually have a little bit higher levels of CD68, which is this marker of microglia activation. So just as I've shown in the first part of the talk, just knocking out BMAL actually causes inflammation in the brain. But in the setting of tauopathy, the mice get really severe neuroinflammation. And the BMAL mice really just don't get this. They have kind of a higher level to begin with, and it just stays at that level. So we're trying to understand how this is, but I think it plays into the role of neuroinflammation in these neurodegenerative diseases, is that it's very much a double-edged sword. It turns out that activated astrocytes and activated microglia can take up A-beta, they can take up tau, they can degrade both species. So in certain contexts, maybe early in the course of the disease, having activated microglia and astrocytes may actually prevent the aggregation of some of these proteins. However, here, later in the disease, when things get out of hand and you have neurodegeneration and too much aggregation, neuroinflammation is known to be a bad thing. And then it starts to cause synapse loss and neuronal damage. And so I think the clock may be a way of kind of changing this barometer of neuroinflammation in the brain and optimizing things. But we still have to understand a lot of that, not just our lab, but the field in general. So in conclusion, we have some initial basic science clues that suggest that the clock, through this BMAL gene, can control several different aspects of neuroinflammation and neurodegeneration. We have signs that BMAL can control redox homeostasis in the brain, that it can regulate astrocyte activation. I didn't show this data, but we think this is through glutathione transferase-mediated mechanism, and that it can also control microglial, daily oscillations of microglial activation through reverb alpha, and that this is a potential druggable target. We also have some data that A-beta aggregation seems to be increased when we fragment the clock and the sleep-wake pattern, but this may have some alternative effect on tau aggregation. So we need better models, and we need to sort of try to integrate all this into what ultimately happens. And it may really depend on the disease and the time at which circadian rhythm disruption occurs. So with that, I want to thank all the people in the lab that helped me do all this work, and all our collaborators, and thank you for your attention. Appreciate it.
Dr. Musiek, thank you very much for another great presentation, and I would like now to open the discussion. I would appreciate if you can come up into the middle, use the microphones, and probably introduce yourself so that we know who's discussing with us. Here it's Dr. Rob Sladen, past member of the Board of Trustees of the Society.
Thanks very much. This has been a remarkable panel. Really want to appreciate that. Wes, I want to express my admiration as an intensivist for what you, Pratik, and the group have done for intensive care for decades, and particularly in bringing people together. I also want to mention to the audience that the passion that you bring to your science, I've had the privilege of seeing you bring that passion to the bedside with your patients. I thought that this came together so well, but I wanted to ask you what you think of adding S to ABCDF and the importance of sleep deprivation, sleep fragmentation, the loss of REM sleep, and the cumulative effects in patients in the ICU, and how we bring this all together.
Sure, and Rob Sladen, thank you so much for the things you said, and you were an early leader and mentor for me in so much of this, so it was really a joy for me to see you out in the audience today. I guess you've not fully retired. I think that sleep is such a huge thing, and I would say over the past 6 months, just 6 months of fresh thinking of all of this, that I'm most excited about the things we're learning about sleep. And so the things that, Eric, that you taught us just now, I just last week read a whole series of around 15 papers by David Bennett and the group at the Russian Memory Alzheimer's Center that they put out about sleep fragmentation. And myself, we have been paying a ton of attention to the sleep fragmentation in our ICU patients, because with the drugs that we give the patients, which are slow-wave sleep suppressive in the unit, the patients are getting less into slow-wave, having more microarousals, and I think we create a playground that generates sleep fragmentation. It's a bad playground, not a good playground. So I was going to ask Eric, too, to teach us more, but we'll get into it. Just the amount of interest in the glymphatics and the clearance of amyloid and tau with better sleep, less fragmentation, and more time. And I was just looking last week at the dichotomous division between less than 7 hours of sleep and more than 7 hours of sleep. I think, it's not a 7-hour magical thing, but it's just striking the amount of accrual of Alzheimer's and dementia when you consistently get less than 7 hours of sleep. So I was wondering, actually, if you could comment on that, and then I see we have another person. But yes, so Rob, we're hugely putting in the S into the bundle. The S, to finish that, the S is part of E. So E is early mobilization and environment. So you don't have to expand beyond 6 letters. Complex multifactorial interventions need to have around 6 as a sweet spot. If you look at complex interventions and mortality reduction, delirium or non-delirium, 6 complex interventions in the bundle is the right sweet spot. So we don't want to go to G and Z, et cetera, but sleep is part of the E environment, and it's a huge piece of our work. But Eric, what are your thoughts about sleep fragmentation and our ICU problems?
Well, I think, it's obviously difficult. One of the most difficult things for me is disentangling sleep and circadian rhythms. They're tied together, they go together, but they're not always the same. You can stay awake and force yourself to stay awake, and your clock keeps ticking. But if you're sleep deprived and fragmented for a long period of time, it can actually disrupt your clock. So we can measure sleep a lot more effectively in a setting like an ICU through EEG, but we don't have great ways of measuring circadian rhythms. So, we're trying to work on that. Blood-based biomarkers for rhythms are starting to come out, but it's a tough. In mice, we can actually manipulate the clock much easier than we can sleep, but in humans, it's the other way around. So, you mentioned the glymphatic system. That's certainly really important. I mean, I think that we're actually working with Mick and Niedergaard's group to see how the circadian clock plays into that. If there does seem to be some component of glymphatic flow. So the glymphatic system, for those of you who don't know, is this glia-mediated kind of garbage disposal system of the brain, I guess, where fluid moves through the brain, and it increases massively when animals sleep. And there's some data in humans now in MRI, but we have no idea what happens to people who are sedated. In mice, actually, sedation seems to increase glia.
Yeah, I know that was a paradoxical finding.
Which is a little strange, but it depends on what sedative you use.
And how you get sedated.
And so, I don't know what happens in people in the ICU. And I think there's a lot of questions. There's a lot of stuff, like the stuff I just showed, that's in mice or in very early days that, the translation of that into the places like the ICU and the dementia clinic is not quite there yet.
Dr. Black?
Yeah, that was a good comment, because actually my colleague Andrew Lim at Sunnybrook works very closely with Bennett and others, and showed the relationship between sleep fragmentation and atherosclerosis and also amyloid deposition. And we also we have a Leduc Foundation grant working with Dr. Niedergaard and Dr. Wardlaw, because we think that the perivascular spaces, that's part of the G lymphatic system. In fact, during slow wave deep sleep, at least in the mouse models, the rat models, it looks like the cells shrink and the sort of perivascular spaces open, and the garbage trucks are kind of getting rid of all your junk from the day, including amyloid. So if you don't get those cycles, then you're not removing, that's the clearance problem, and also that Dave Holtzman and Bateman showed several years ago on a 24-hour cycle. The problem with aging is clearing amyloid, it's not overproducing it. So this is all a very hugely important orchestration of, the 24-hour clock. And, maybe microglia wake up too, I don't know, but it seems that there is this fluid flow. And so if you have perivascular spaces that aren't working because you've got atherosclerosis or you have venous collagenoses, then you're not going to be, your roads are not working. So even if you're sleeping, probably you're not getting ready. But I think sleep is now starting to be really on the radar in the context of dementia and aging, and now we can monitor it so much better, right, with different devices, so that you can really know what people are doing. We're doing home sleep testing in mild cognitive impairment and Alzheimer patients just to see what's happening to them, whether they're snoring or not. But that's another thing. Compliance with CPAP is very problematic. So I think it's actually quite an important new development in our awareness of what happens. There's also, interestingly, you may not know this, as you age, there tends to be trends in younger people and older people for you to do better cognitively. So older people do better cognitively in the morning. Young people do better cognitively in the afternoon, and they can actually vary quite a bit. So when you give tests to 25-year-olds in the morning, they don't do as well as they do later in the day. Also, sleep is cognitive enhancing. You can have people do motor learning tasks and then look at how they do when they repeat that before and after a nap or an overnight sleep, and they do better, when they've had sleep. So I think there's a lot going on in sleep that we don't fully understand. But the other thing I think is really interesting that you're pointing out is this kind of interplay between amyloid and tau that are both doing something good, something protective, and then something bad if they get out of hand, or we just don't understand. We've really got to understand. We really have to appreciate more what these responses are. I mean, amyloid is made in the rest of the body too. It's not just in the brain. So we may be playing with fire and just trying to remove it. So there really are this idea that it might be. I mean, we know that amyloid and tau are probably co-partners in pruning, in development. So amyloid hits the synapses, tau cuts the axons as you shape what you're going to learn. And if you don't, or by your environment, and if you don't prune, you end up with autism or Asperger's or schizophrenia. So I mean, it's really important for us to understand the role they play and then how it goes afoul in later life.
Thank you. Christine Engelhardt?
Oh, thank you very much for these three fascinating talks. I just have one question. Is this BMEL somehow associated with melatonin, and how, if yes or no, would that be any drug you could give your patients to regulate the sleep?
Yeah, so the SCN actually signals to the pineal gland to regulate melatonin secretion. So melatonin is kind of, you can measure melatonin as kind of the gold standard of someone's circadian phase. So yes, if you knock out BMEL in the whole animal, they lose their melatonin rhythms. If you knock out BMEL in the pineal gland, they'll actually lose their melatonin rhythms. So there has been some work, you may know more about this, and there's been some studies in Alzheimer's disease with melatonin and early light therapy that have been kind of a mixed bag of studies. I don't know how well any of them have been done. Some of them look more promising than others, but people have been thinking about that for a while. The problem is the responsiveness to melatonin can wane in older people, and so it may be that you just don't have the receptor signaling left anymore to respond.
Yeah, I think the trials of melatonin have been, they haven't really led to any definite therapy. Sometimes we use it, we offer it to people, and they try it out, and sometimes it helps and sometimes it doesn't.
Now, in your intensive care facilities, how do you use it to sedate patients then?
We've switched from a lot of heavy GABAergic use to either analgo sedation with just fentanyl only or morphine only, or no sedation policies where patients are on the ventilator but not getting sedatives unless they request it. If somebody's bucking a ventilator and desaturating and has double-digit high peeps, then we use some sedation. But usually we use an alpha-2 agonist or a narcotic only.
And then ultra-early tracheostomy for them?
Very few of them get tracheostomy. Most of them are getting awake and alert and walking around and off the ventilator. But if they, if they are approaching double-digit days on the ventilator and no sign of coming off soon, then we'll trach them for comfort. But the trach rates have plummeted since we started doing the 8F bundle. We trach a fraction of the patients that we used to trach 10 years ago because they're getting off the ventilator so much faster.
All right. Dr. Bev Arthur, fellow Board of Trustees member.
Right. Thank you for a wonderful symposium. I remember I too work at Sunnybrook, and reaching out to Dr. Black to have coffee after being up all night on call, and she said, "You know, Bev, you're making amyloid right now," which explains a lot. Anyway, thank you. My question, two questions, Dr. Black, Dr. Vasili. Can you tell us, Dr. Black, where you think the imaging field might move in the post-op perioperative period? Perhaps not delirium, but in this post-operative neurocognitive deficit field. And then I just want to pick up one more question about dexmedetomidine and its current use in the ICU and your recommendations.
We'll come back to that.
Well, maybe you can tell me how often people get imaged, when they come in with, say, a body problem or sepsis in terms of what's going on in their brain. I think you would need you would need to know what their brain looks like, just like maybe how their heart's doing when they're in this kind of situation. And I don't know how routinely that's done. Whether they have severe white matter disease, whether they have microbleeds. I mean, it will tell you a lot about the state of that brain, not just infarcts. I think in North America now, with at least many jurisdictions, reasonable use of antihypertensives and vascular risk management, we are seeing people that have fewer of the pathologies that we used to see. Even the white matter hyperintensities are kind of less frequent than they used to be. But I think that just having kind of a brain screen where you're looking at hippocampal volume, you're looking at the extent of the white matter hyperintensities, or the pathologies would be part of the way that you're kind of evaluating how they're doing. But I don't know if that's done at all.
Not routinely. It's more for just clinical indications, I think. If somebody has something clinically indicated. We're going to be doing that in brain too.
Because I think it would be interesting to see how predictive that is, or whether you can, because I think we're in the field of dementia, we're getting very interested in what we call brain resilience, which is a combination of things we don't understand. But still, some of it is, it's almost paradoxical, because sometimes people who are very high functioning and seem to have a lot more reserve will have a more atrophied brain at the time they actually present with symptoms. So they've been fighting the amyloid and the tau. Eventually it wins, but...
We did volumetrics, and DTI work on patients who had delirium and didn't have delirium, and we found that delirium duration, just like it was a predictor of death and length of stay and cost of care and dementia, delirium duration was a predictor of greater degrees of atrophy in the dorsolateral prefrontal cortex and in the hippocampus. But we didn't know cause and effect. There's no chicken and egg proof there because you didn't have the evaluations beforehand, but it was an independent predictor.
It's interesting.
And your second question was about dexmedetomidine?
Right.
Yes.
Can I just make a comment? So
Can
on
you-
the brain imaging-
Better come to
Yeah,
the mic.
sure. So
Yeah,
sorry,
Rob
Bev.
Sanders.
Yeah,
Go ahead.
I'm Rob Sanders. Just a comment, the Neurovision study was preliminary data from that came out at one of the big stroke meetings about 3 months ago. I wish it wasn't. And go closer. Sorry. And the finding from that was about 7% of people had a post-operative covert stroke for patients who had undergoing major non-cardiac surgery requiring at least a 2-day stay. These were new DWI identified lesions. The scanning occurred day 2 to day 9 post-op. It was a cohort study of 1,100 patients. And it was associated with a increased incidence of a 2-point drop on the MoCA at a year. So there is a lower incidence of perioperative covert stroke in that non-cardiac population, which is, substantially lower than the carotid endarterectomy and cardiac surgery story. But that does have some long-term cognitive implications that would appear in that preliminary study.
I think the microinfarcts also, I don't know if that include microinfarcts, because sometimes they're very tiny. Might be important and may relate to paroxysmal atrial fib, that's happening too.
Well, I'd like to challenge that. I don't think 7% covert stroke is
Trivial.
trivial, is nothing. I think it's upsetting. If we would have 7% myocardial infarction that is sort of covert, the whole cardiology society would generate billions of dollars to go to research for that. So I think 7%, there is publications which are non-cohort, which are prospective, that show 10% covert stroke. I think it's dramatic and requires immediate reaction, immediate, beyond the perspective and prognosis of then maybe triggering dementia or so forth. So I think thank you for the discussion, but I don't think it's trivial. Then I misunderstood. Well, you didn't say trivial, but you said it's a minor number, and I don't think so.
Small lesions which are observed. So it's all contextual. This is a single, single study. Do I think that this is something which should be ignored? Absolutely not.
Okay. Well, thank you very much for clarifying that.
Okay, I think she had one more question.
Right, I think just to comment out dexmedetomidine and where that sits in the field, current use.
Okay, just a general comment. Okay, so first of all, the theory was that alpha-2 agonists hitting at the locus coeruleus would be a better way to go than the VLPO with GABAergics and such. And we don't know if that would be mediated by sleep or what it would be mediated by, anti-inflammatory effect, et cetera. But the bottom line is that if you compare alpha-2 agonists to GABAergic drugs, whether it be MINS in JAMA, Prodex or Midex, both in JAMA, that's 3 studies, SEDCOM in JAMA, that's 4 studies of a couple of thousand patients. Every single time, the patients who got exposed to the alpha-2 approach of sedation had either less delirium, less time on the ventilator, better communication to nurses. Everything looked in favor of this approach. Was it because they were not being exposed to something more dangerous? Or was it because they were being exposed to something that was actually advantageous in the alpha-2 agonist approach? I don't know that. We don't know that in medicine. But what I can say is that it's a fairly consistent message. You could add Michael Reed's study in JAMA too. That's a 1, 2, 3, 4, 5. It's like 6 studies already in JAMA now. His study was of alpha-2 agonists in delirious patients being kept on the ventilator, and they got off the ventilator sooner. So the bottom line for me as a clinician at the bedside is that internationally, repetitively, in a consistent manner, I think that we now know that an alpha-2 agonist approach or a narcotic approach without heavy GABAergic use seems safer and better for humans than a tried and true older approach, which is either heavy benzos or propofol. We just completed MENS 2, which was propofol versus dexmedetomidine. Pratik Pandrapandi and Chris Hughes are the PIs on that project, and others in this room have participated. And we haven't analyzed the data, but we are analyzing the data this summer, and we hope to be submitting MENS 2, propofol versus dex, in the fall. So I think that I, as a clinician, tend to shy away from heavy sedation to get patients comfortable. I tend to do it either with analgo sedation by itself or with an alpha-2 approach. Or, if I don't want to use a drip, I will use antipsychotics. I'm not using the antipsychotics to treat delirium because I just showed you the Mind USA study. But I think antipsychotics do create a sedative effect that doesn't suppress the respiratory drive. And so if I have a patient in a less ICU sickness state who isn't going to be subjected to a drip, I can use intermittent drugs of antipsychotics for a day or so and get by with allowing them to tolerate a BiPAP mask, for example, in COPD or CHF, et cetera. So those are our approaches to sedation now. Thank you for asking.
Can I just ask quickly about anticholinergic drugs and sort of
Anti-what?
anticholinergic effects? Like, to what extent is that playing into all of this at all?
Well, acetylcholine depletion or dopamine excess were for a long time thought to be the hypothesis of the neurotransmitter hypothesis of delirium, but that hasn't panned out in a beautiful way in any single intervention. We definitely, as part of the C, choice of drugs, we definitely try and shy away from anticholinergic drugs when patients are delirious. So 100% we support getting away from anticholinergic drugs, but it's not as though anticholinergic levels, the Larry Toon stuff, none of that stuff is panned out in a kind of a crisp way.
All right. I would like to close the session in thanking you, Dr. Black, Dr. Eli, Dr. Musiek, for devoting your time with us and sharing your knowledge, educating us. It's always good to have a competent anesthesia background scatter. You may not know when you need it. So you really moved us forward. Thank you very much.
Thank you.
And on behalf of the IARS, I wish you a good evening in Montreal. Thank you.